All right, so we are here to talk about viruses. Fun stuff. And the first one is I can list and describe the parts of a virus. So let's draw that out really quickly. Y'all know my drawing skills, so don't get too excited. Excuse me. So, and I'm just going to draw a random virus. Viruses come in all different shapes and sizes, but I'm going to draw something that's within my capability. So a virus is going to be surrounded by a protein coat, protein shell, sometimes you'll hear it called, and that is called the capsid. Okay, and all viruses have that. Inside that capsid is some kind of genetic material. That genetic material can be DNA, or it can be RNA, and it can be double-stranded, or it can be single. I don't know why anyway, I could just put the vowels, but whatever. Okay, and it can be double-stranded DNA or single-stranded DNA, and it can be double-stranded RNA or single-stranded RNA. So it can be either one of those. Some viruses then also have a lipid membrane around them, and a lot of times they get this from the cell they escape from. And so it's a phospholipid bilayer just because it got it from the cell. And if it has that, that is called an envelope. And not all viruses have that. So all viruses have the protein shell, they have the genetic material, those two parts, but not all of them have an envelope. And so that says I can list and describe the parts of a virus. All right, I can explain the lytic cycle of a virus. The lytic cycle, lytic comes from the word lysis, which means to burst. And so the lytic cycle is going to be one that's going to end up bursting the host cell and killing it. And it has 5 steps. The first step is attachment. So the virus is going to attach to the cell somehow. There's generally some kind of receptor or something that they can use to attach. The second is entry. And so it inserts its DNA inside the cell. The protein is going to stay outside. The DNA is going to go in. The third step is synthesis. Synthesis means to make. And so this is where the virus is going to turn the cell into a factory, and it's going to tell the cell to make more genetic material, whether that's DNA or RNA, of the virus, so make copies of the virus's genetic material, and it's going to have transcription and translation occur to produce the proteins to build that capsid or that protein shell. So synthesis is where it's just making all of the parts, and then assembly is where it's going to put all those together. It's going to assemble the viruses. And then it's going to go through release. And release is where it's going to burst or lyse that host cell, and all those new viruses are going to come out. So that's the lytic phase, or the lytic cycle. And it's called that again because it bursts or kills the host cell during that release stage. The next is I can explain the lysogenic cycle of the virus. Okay, so here's your lytic on the left, okay? Attachment, entry, assembly, or synthesis and assembly, and then release. Lysogenic means, genic comes from Genesis, lyso, lytic, or lysis. So this is a cycle that can genesis or create, that's what genesis means, to create a lytic cycle, okay? But it's not going to be lytic in and of itself. With the lysogenic cycle, the first 2 steps happen, okay? Attachment and entry. So the virus attaches, it inserts its genetic material into the host cell, so there's that entry. But instead of going through synthesis, assembly, and release, it cuts the host DNA, inserts its DNA inside of it, and then it just kind of sleeps there. So every time the cell replicates, and this is shown in bacteria, but it would do the same thing in you. Every time it replicates, it's going to copy the host DNA, but also that viral DNA because it's now a part of the host DNA. And so the whole purpose, the whole idea behind biological life is to survive and reproduce. And what that ensures is it ensures that your genes get passed down. And we'll talk more about this in the evolution unit. So a virus is doing that, whether it's doing lytic or lysogenic, it is getting its genes passed down. Now, in our mind, we think, oh, well, it needs to exist as a virus out in the atmosphere. It needs to get passed from host to host, but it really doesn't. If it is sleeping, so to speak, in stealth mode in the organism's DNA and your DNA or this bacteria's DNA, and every time that bacteria divides or every time your cells divide, it gets copied, then it's doing what it's supposed to do. It's passing its genetic material down. Is that genetic material cloaked in a protein, like the virus over here? No, but it's still passing that genetic material down. Now, the thing about the lysogenic cycle is that at any time it can revert to lytic. So it can be sleeping for years, weeks, days, it doesn't matter. And then all of a sudden it can kind of wake back up and go into that synthesis, assembly, and release. So it can convert back to lytic. We see this with chickenpox a lot. Maybe not so much with y'all because you got the vaccine, but those of us who had chickenpox as a child, it inserted its DNA into our cells and it sleeps for years and years. And for some people, it never wakes back up. They never get shingles. But for other people, it wakes back up and goes back into the lytic cycle and it expresses itself as shingles. So that is the lytic cycle, the lysogenic cycle. And it's called lysogenic again because it can create that lytic cycle. It's just not actively in that phase when it's just lysogenic. All right, and then I can compare and contrast the lytic and lysogenic cycles. So the lytic cycle, well, let's go with both first. They both have that attachment, those first two steps, and entry, and they both lead to genetic material being passed down. Okay, so whether it's doing it in the form of new viruses or whether it's doing it in the form of just as host DNA, as being incorporated in the host DNA, they're both passing that down. The lytic has those last three steps. It's going to synthesize the genetic material and protein. It is then going to assemble new viruses. And then it's going to release them. And the host cell dies. Most of the time. There are some, and I don't think they're considered lytic. Because they don't lyse the cell. I'd have to look it up. But there are some that actually bud out. I think HIV does this. It buds out new viruses, and so it doesn't always kill the host cell. It kind of takes over it, so it might as well be dead. But all right, and then the lysogenic, let's see, viral DNA incorporated, put into host DNA. And that's called prophage when it's like that. Or I think sometimes it's called a provirus. And it is dormant is probably the best way to describe it because it's not actively in that lytic cycle. One thing I want to explain about a virus is viruses generally, the DNA or the RNA that they have simply gives instructions on how to make more viruses. So they don't really have a purpose. I guess they do kill things, which keeps population under control. It's a weird purpose, but they don't really have another job. Like literally they infect you and then they tell you how to make more viruses. And then those viruses infect you and tell you how to make more viruses. It's not like they have some contribution to society that they're doing in addition. They don't have extra genes in addition to. Their genes literally say, here's how to make more viruses. So they're just kind of a nuisance, really, which y'all know that because nobody likes viruses. All right, the last I can statement is I can explain how a retrovirus works. Okay, so a retro means to go back. So a retrovirus is an RNA virus, okay? That RNA goes through reverse transcription. So remember, transcription is going from RNA to DNA. From DNA to RNA. I'm thinking of what I'm doing here. It's going from DNA to RNA. They're going to go through reverse transcription using an enzyme called reverse transcriptase. Good name for it. And that's going to take that RNA back to DNA. And then they'll go through transcription. Didn't make my line long enough. And then translation. Okay, so that's really all a retrovirus is, and they're called that because they have this extra step where they have to go backwards, so to speak, back to DNA before they can follow central dogma. Okay, and I say this is all happening your cells or the cells that they're infected. So one of the things they do is they have a gene that tells your cell to make reverse transcriptase. Your cell makes that reverse transcriptase that then codes this back to DNA, and then it can go through transcription and translation to make new copies copies of the protein, but also to make new copies of the RNA to be packaged in that. And so that's what a retrovirus is and why it's called that. So I hope that helps. If you've still got questions about viruses, let me know.